Okay, so today we will be starting with the proteins. And to start with, first of all, you know that proteins are found in different sources of food, right? So would you like to name any source of food in which proteins are present?
Egg, meat.
Very good, exactly. So for the sources, you can say you have egg, meat, other forms of poultry, and dairy products. So all of these, they consist of the proteins. So what happens is that whenever we consume them, they go inside our body, and also in the body, we say that there are different types of proteins present. Like some proteins are needed to make new tissues in the cell, or to just repair the damaged cells and tissues. So similarly, we need protein so that we can perform all of these metabolic processes. And also there's one more thing about the proteins, which is that they are quite essential in a way that they can you can say they are the building blocks of different molecules, like the hormones, the antibodies, and also like the enzymes. So all of these things, they are made up by the proteins. Now, first of all, protein is a macromolecule. So a macromolecule here means that, like, let's suppose if I'm talking about any protein and it consists of simple monomers like this would then be considered a macromolecule. Now, if you remember that we studied about the different groups, like we studied about the amino group and also the carboxyl group. So what happens is that in the proteins you have these amino acids, and each monomer, you can say, is an amino acid. Now remember that you have at least 20 amino acids that are different in nature, like in chemical nature, that are present in our body. So this means that maybe this could be an amino acid one, that could be methionine, this could be lysine. So there are different amino acids that are present in the body. However, like you would know that for the basic structure for an amino acid, like let's suppose, let's just draw the basic structure of it. You would see that it consists of a carbon, and along the carbon there is an H, that's the hydrogen that's bonded to it. And as you know, do you know how many bonds can this carbon form?
4.
Very good, exactly. So, one is attached over here, the other one, and like this, there would be 4 bonds formed. Now, the other 3 bonds, like one of them is bond with an amino group, as I told you, that's known as it consists of nitrogen along with 2 hydrogens bonded to it. And then we also have a carboxyl group, which is something like this, that has, if you remember, the double bond with the O, that's known as the carbonyl group, and this is known as the hydroxyl group. And lastly, you have an R group over here. Now, this R is known as the side chain of an amino acid. This means that the side chain varies in all different, 20 types of amino acids. Like for instance, in some amino acids, instead of, it could be a hydrogen bonded over here, then this would be known as methionine or methionine. But like if I say that over here, I place a methyl group is something like this. So if I place it over here, this would then become a different amino acid. So the overall structure would remain the same, but only the side chain, which is over here, the R group, would vary. So is it clear? Any questions till now?
Yeah, it's clear.
Okay, very good. Okay, now if I say that, if we just move go back to this example, that this protein consists of so many amino acids connected to each other in a chain. So each of the bond over here It forms again by the process of condensation. And now you would see how would this happen. Now, like, let's for instance, if we take two amino acids together and we have to form peptide, because a peptide is something that consists of like two amino acids or sometimes three too, but again that varies, and whenever you have more number of amino acids, that is known as a polypeptide. But like, let's just start from the scratch, and if we have two amino acids, let's draw their structure. This is one. So this is the carboxyl group, this is the amino group. You have an H over here, and the other amino acid, again, it consists of another side chain. It could be the same as well, but let's just suppose it's a different amino acid. And then we can have the amino group again over here, along with one more carboxyl group and an H over here. Now what happens is that you are going to have the process of condensation, which means that a water molecule is going to form. And how it's going to form is that the H from here is gonna come, and it's going to react with the OH of this group. And this would be released, and this would form a water molecule like this. And the remaining amino acid that they're going to now join together like this, and this would form a you can say a dipeptide maybe, or we can call it a peptide as well. And you would put the bonds like they were before, similarly, and over here as well. Okay, now the main thing over here is that this link that formed is known as the peptide linkage. And the peptide linkage, it consists of a nitrogen, this hydrogen that's bonded to it, along with the carbon that is bonded to a carbonyl oxygen. So, like, is it clear now? This is known as the peptide linkage that's found in the proteins.
Yeah.
Okay, now apart from this, we have also, let's see the different types of amino acids, as we just know that. So first of all, the 20 amino acids, they are divided into two types. Okay, so do you know the two types of the amino acids, how we categorize them?
I think I know one. It was globular, right?
Okay,
Or no?
yes, you can say that as well. That's another classification in which we say that okay, so over there you say that there's a globular protein and a fibrous protein, and that's related to its solubility and structure. But if we just go to the basics, and from the simplest molecule, that's the amino acid. So in that classification, we have two types that's on the basis of the R chain or the side chain.
I'm not sure.
Okay, so that's known, like, you have polar amino acids and you have non-polar amino acids. So now, just remember that a polar amino acid would be any amino acid that can easily dissolve or be soluble in water, while a non-polar amino acid would be something that would not, dissolve into the water. So this is the basic difference between them, and like this, they actually differ.